Up the Down Staircase. Telencephalon. Part 1. The cerebrum, or telencephalon, is the largest part of the brain that includes the two cerebral hemispheres consisting of an outer layer of gray matter called the cerebral cortex, an inner mass of white matter, large masses of the gray matter lying in the depth of the white matter and called the basal ganglia or basal nuclei, and a cavity called the lateral ventricle. The basal ganglia include caudate nucleus, lentiform nucleus consisting of two functionally distinct parts, the putamen and the globus pallidus, amygdaloid body or amygdaloid nuclear complex, and claustrum. The caudate nucleus and the lentiform nucleus together form the corpus striatum, that consists of two functionally distinct parts. The caudate nucleus and the putamen form one unit called the striatum or neostriatum, while the globus pallidus, that is divided into external and internal segments, forms the other unit, the pallidum, or pallostriatum. Phylogenetically, the amygdaloid body and claustrum are archistriatum. About 25% of neurons in the striatum are medium-sized GABAergic ones with extensive spherical or ovoid dendritic fields. They are called medium spiny neurons or spiny projection neurons. They receive inputs from the cerebral cortex, thalamus, and pars compacta of substantia nigra, and they send axons to globus pallidus and pars reticularis of substantia nigra. Neurons in the globus pallidus are large, with dendritic arborizations that have the shape of flat discs, lying parallel to one another, parallel to the border of the nucleus, and perpendicular to the afferent striatopallidal nerve fibers. The latter form a part of the striatopallido-nigral pathway that traverses the globus pallidus and gives it the pale appearance by which it is named. Neurons of the globus pallidus internal segment send axons to the thalamus and to pedunculopontine nucleus, while neurons of the globus pallidus external segment send axons to the internal segment and to the subthalamic nucleus. That in turn gives excitatory output to both segments of the globus pallidus and pars reticularis of the substantia nigra. Due to their multiple connections, striatum and pallidum perform various functions. They take part in the planning and programming of movements, influence the motor cortex via the thalamus to provide the final movements through the corticospinal tracts. Moreover, the globus pallidus internal segment projects to nuclei in the brainstem and spinal cord. The caudate nuclei, due to the interconnections with the frontal parts of the neocortex, also play a role in some cognitive processes. The amygdaloid complex is a group of more than 10 nuclei that are located in the mid-temporal lobe. These nuclei have intranuclear and internuclear connections and extensive connections with cortical and subcortical regions. The following main types of neurons are described in the nuclei of the amygdaloid complex. Pyramidal-like cells, number 1, with 3 to 7 spiny dendrites. Small stellate cells, number 2, with 2 to 6 spiny dendrites. Medium-sized cells, number 3, that have an ovoid or fusiform soma and 3 to 5 spiny dendrites. Medium-sized ovoid cells, number 4, with spiny, largely bipolar dendritic trees. And large cells, number 5, with very long, thick spiny or aspiny dendrites that travel to other nuclei. The amygdaloid complex plays a primary role in the processing of memory, decision-making, and emotional responses, including fear, anxiety, and aggression, and it is considered a part of the limbic system. The claustrum is a thin sheet of neurons lying between the insular cortex and the striatum. It is reciprocally connected with almost all cortical areas, including motor, somatosensory, visual, auditory, limbic, associative, and prefrontal regions, and receives neuromodulatory inputs from various subcortical structures. The claustrum contains two major classes of neurons: projecting neurons, with spiny dendrites, and local interneurons, with aspiny dendrites. The majority of the neurons are glutamatergic, while a smaller proportion of cells, from 7 to 12%, are GABAergic. The claustrum takes part in sensory processing, sensory motor integration, and is thought to play a role in the segregation of attention. The cerebral cortex, or pallium, regulates language, thought, and behavior via sensory, motor, and cognitive processes, that is, performs the high nervous activity. Taking into account its structure and functions, the following main types of neurons of the cerebral cortex are distinguished. Pyramidal, have the shape of a pyramid, the top of which is always directed towards the surface of the cortex. One dendrite arises from the apex, extending through the entire thickness of the cortex, and other dendrites arise from basal angles. The axon arises from the base of the cell, establishing numerous synapses in other regions of the CNS, subcortical projection neurons, or cortical within the same hemisphere association neurons, or opposite hemisphere commissural neurons. The pyramidal cell axons give off recurrent branches before leaving the gray matter. All pyramidal neurons are excitatory, using glutamate or aspartate as transmitter. According to the size of their cell bodies, pyramidal cells are divided into small, about 10 micrometers, medium, about 50 micrometers, and large, from 70 to 120 micrometers, the latter in the primary motor cortex being called pyramidal cells of Betz, or Betz cells. Stellate, or granule neurons, small, from 8 to 10 micrometers, star-shaped neurons with numerous dendrites of approximately the same length, which extend chiefly towards the surface of the cortex, but in some cases may be oriented in different directions. Horizontal cells, or cells of Cajal, multipolar spindle-shaped neurons providing interneuronal connections. They are located in the superficial cortical layers. Fusiform neurons have a fusiform cell body with their long axis being vertical to the surface. They are located in the deep cortical layers. Neurons of Martinotti, small multipolar cells with an ovoid or polygonal body and short branched dendrites. The axons form contacts with the pyramidal cell dendrites. Chandelier cells, GABAergic interneurons forming extra-axonal connections that intertwine the initial axon segments of pyramidal neurons. Their name comes from the specific shape of their axon arbors, with the terminals forming arrays called cartridges. Basket cells, multipolar interneurons whose axons surround somas of pyramidal neurons, forming baskets. Double bouquet cells, GABAergic interneurons that have tightly interwoven bundles of long, vertically oriented axonal collaterals resembling horse tail. In the August of 2018, an international group of scientists discovered new type of inhibitory GABAergic neurons present in the molecular layer of the human cerebral cortex. They exist in humans, but have not been reported in rodents or other animals. These cells were called rosehip neurons because their axon terminals targeting dendrites of pyramidal neurons in layer 3 resembled the rosehip berries. To be continued.